Okay, so I'm going to follow up from where Michael left off. So Michael gave you a terrific synthesis of what we have put together over, honestly, the last more than 20 years. And what I'm going to try and do is walk you back through parts of it and focus on a couple of features that go along. And, I'm a little uncomfortable being up here on a stage with all these chairs, and I keep expecting Clint Eastwood to come in. So I'm a little anxious about political problems. Oh my God. Hold on, I think we do have a political intercurrence here. So I need to remind you, just for one moment, what April 15th did for you. And one of the things it did for you, aside from ushering in the sequester, was to foot the bill, even if transiently, for a 200-bed referral hospital that you own, you can admit to. Maybe a little indirectly. But it is on a 32-acre enclosed campus in Bethesda, Maryland, where we have medicine, pediatrics, intensive care, and surgery, and where we perform retrospective review of research. And there are a lot of great ways to be involved with the NIH, and you need to know about them because we've got opportunities for you and your fellows and your students. And those opportunities include post-baccalaureate opportunities for your children like mine who get to the end of college and don't know what to do. There are great opportunities to learn about science and to become investigators. Medical student rotations for those students that really are exceptional in your program who want to learn more. And then we've got residencies, we've got fellowships, and we've got rotations for people to come there for prolonged periods. And there are advanced programs like the one that Michael is in, such as this medical student research program, which is 1 year of paid research to really focus on clinical activities. There are great opportunities for young faculty members. So if you're a fellow or you're just ending fellowship and you're looking for opportunities to pursue science and medicine together in one laboratory, these assistant clinical investigator pathways, Statman fellowships for basic investigators, and finally, not that anybody would ever want this, but 12 years of covered support for the Lasker Clinical Scholars Program. 12 years, 5 years intramural, 7 years extramural. You can really do things. And who are the giants that walk these halls? Let me just show you the actual people who take care of actual patients in the actual National Institutes of Health. Tony Fauci, Dan Kastner, Neil Young, people that you know, people you refer to, people that you actually think are good at what they do. These are the people that are on the floors and in the clinics at the National Institutes of Health. So think about this place when you're thinking about where to train, what to do, where to send your best fellows, where to go yourself for fascinating opportunities in science and medicine. Phew! Okay, well, so Clint is gone, but I need to tell you about these things because if I don't tell you, who's going to tell you? So this is someplace you need to know about. Now, today we're here to talk about GATA2, and we're talking about GATA2 because it's new and it's important, and I promise you've seen much more of it than you think. So we'll work through this pathway, and I promised Jordan we'd be done by 1. So here, you see GATA2 right at the center of all these things. How does it do the things that Michael talked to you about? It's by being at the center of an enormously complex set of pathways. Keep in mind that down here are transcription factors, here are HDACs, here is endothelial nitric oxide synthase, angiopoietic factors, as well as many other genes that GATA2 regulates directly and indirectly throughout. So the best way I know to understand these things is to look at individual cases and to build up from there. So I'm just going to walk you through somebody who's very emblematic of this problem. So this woman who actually was in the hospital last week for a routine 18-month post-transplant check had a perfectly normal childhood until she was 12 years old when she had severe chickenpox that kept her out of school for 3 months with a prolonged hospitalization. Following that, she was normal again, but when she was 17, she was noted to have neutropenia and lymphopenia. At 23, she developed her first episode of severe HPV infection, genital HPV, leading to multiple surgeries, fulgurations, and procedures. At 26, she was given the diagnosis of all people who have complex diseases that are not understood, lupus erythematosus, and was managed with some of the less toxic drugs that are available for that syndrome. At 28, she developed fever and leukopenia, and later that year had a splenectomy for the leukopenia, which, to the surprise of all, showed that to be full of mycobacteria. Finally, during that hospitalization, she had severe C. difficile colitis with mesenteric node involvement and what they thought was mesenteric ischemia and thrombosis, leading to long-term anticoagulation. At age 33, she came to see us with this problem, which you can see here, this large mass that is draining out of her right thigh into her right buttock. At that time, her white count looked like this. Michael told you about some of the cytopenias. Well, she really had all of them at this point. Remember, she's 33 years old by now, and she had gone through periods of life when she was perfectly normal. So she was leukopenic, anemic, had a normal to high platelet count, but you can see the other cell lines were down. Her marrow was important to think about because it was hypoplastic with mild dysplastic changes. And you see here an example of the hypoplastic marrow in young people. If you see hypoplastic bone marrow changes in young people, this disease has got to be at the top of your differential. It's absolutely critical that all hypoplastic cases be at least considered for this. There are certainly other diseases in the differential, but you need to think about this. Despite the fact that she had no monocytes in her peripheral blood, she had plenty of macrophages in her tissue, as you see here on CD68 staining, and despite the fact that there were no B cells in her peripheral blood, she had plenty of plasma cells on peripheral blood staining, and her immunoglobulins were normal, which reminds you that for a long period of her life, she made B cells and she made monocytes and she put them in the tissue, and they just are sitting there, waiting for reactivation, waiting to do something, waiting for Godot. So she came to see us in July of 2009 and was doing fine. Really, we'd cleared up her infection using standard drugs, and she did okay otherwise. And then in 2011, during what was supposed to be a family vacation in Durango, she became weak and went to a local emergency room where they noticed that she had 5% blasts in her peripheral blood. That is considered not good. And very shortly after that, during the same vacation, became acutely aphasic and wound up in an emergency room where she was found to have these problems here. You can see here a large stroke on her left side leading to her aphasia. A second one came up a few weeks later that was on the right side, which caused her to have some hemianopsia. And this was explained by vegetations on her mitral valve. So at that time, of course, now she's got blasts in the peripheral blood, and she's got a marrow that looks consistent with that. She's hypercellular. She's diagnosed with refractory anemia with excess blasts and has monosomy 7 on her cytogenetics and winds up needing urgent chemotherapy and bone marrow transplantation. I tell you this story because it's really emblematic of this disease, going from completely normal to not so normal to acutely and desperately ill. And what would the marrow look like in this setting? Now, again, I don't know if you have a program like ours where, usually people just send you back the report on the marrow, and it's very hard to get a look at what they actually saw. But I'm very lucky to have a collaborator, Kathy Calvo, who, shares primary data with us. And the difference in the marrows between these people are shown here. In normal marrows, the granulocytes are very abundant and have normal granulation. Monocytes are easily detectable, as are lymphocytes and blasts. In this patient, what we would have seen are hypogranular neutrophils, shown here. Monocytes are gone. Lymphocytes are gone. These are mostly all the B-cell precursors have disappeared. And blasts are increased. So the reason I show you this woman is that she encompasses all the features that we're talking about here. Severe herpes virus infections, cytopenias, severe papillomavirus infections, bacterial susceptibility, thrombotic problems, as well as then moving toward hematologic malignancies. So, as Michael pointed out, there are a lot of things that are features of this disease. It took us a long time to put all these together. We saw our first patient with this disease in 1992. We published our first paper describing it in 2010, reminding you that NIH is often thought of as being not in a hurry. And the features of it turn out to be, once we finally got to the gene, described by the influences that are shown here. So let me just remind you for a minute about GATA factors. If they're not a standard part of your vocabulary, they certainly were not of mine. There are 6 of them. And they break down into two major groups, the hematopoietic factors, GATAs 1, 2, and 3, and the more somatic factors, especially involved in cardiac function. GATA1 is involved in the anemia and thrombocytopenia that you often see associated with myeloproliferation in Down syndrome, but there are also cases of Diamond-Blackfan anemia that have been caused by GATA1. GATA2 is what we're talking about. GATA3, despite what the lymphomaniacs will tell you, the human phenotype is only around hypoparathyroidism, deafness, and renal disease. But then in GATAs 4, 5, and 6, you can see a strong flavor for cardiac involvement and pancreatic involvement. Now, you want to just keep in mind that the GATA factors are characterized by these two zinc fingers that are central, that do the DNA binding, and then the aspects of variation on the C and N termini, which are presumably involved in how it interacts with other transcription factors to bring them into the different regions to activate transcription. So one of the things that you'd predict from this is that since the binding sites are relatively similar, there could well be a fair amount of overlap between what GATA2 does and binding sites that might be more devoted to GATA1, 3, 4, 5, or 6. And I think in part this explains why we see much overlap with some of the endocardial and the ototoxic effects of the other GATA factors. Now, as Michael pointed out, this gene comes in 7 exons, and these mutations were initially reported by the group of Hamish Scott and Marshall Horowitz to be autosomal dominant inhibitory mutations. And they thought this because many of these mutations are in fact protein positive, and they might be expressed and might be expected to inhibit function of the normal remaining allele. All cases that have been identified so far are heterozygous. The homozygous knockout mouse is lethal. And so the protein positive mutations might be dominant negative. However, when we started sequencing more through these and identified that many of them had changes that were deletions or insertions that could not make protein, it became clear that the only mechanism that would unify those two species, that is the protein positive and the protein negative, would be haploinsufficiency. Despite that, as we went through these, we found several families in whom we could not identify mutations. And so in searching through those, we went back to try and understand the early literature on GATA factors and what they did. And the work of Emery Bresnik is really quite enlightening here. And what Emery had done was to look at all of the GATA binding sites in the genome. And just keep in mind there are 1.7 million GATA binding sites in the human genome. And out of those 1.7 million, almost all of them are either intronic or in enhancer regions. And you can see about 7% are in the regions that we normally think about when we sequence through DNA, promoters, 3' UTRs, and so on. So looking back then at the broad spectrum of where GATA2 might bind, Amy Sue in the lab, who has really run the genetics of this project, said, well, wait a minute, if we've gone through all the exons and not found anything, maybe we should go back and look at other conserved elements. And so using what's called the Genome Evolutionary Rate Profile, or the GERP score, she went back to say what other regions are preserved. And here what you see is deep in intron 5. Here deep in intron 5 are two highly conserved elements. And the black box reflects things that are completely conserved all the way back to zebrafish. And so what you see here is a 28-base fragment that is completely conserved. It contains an enhancer element as well as a GATA binding site, GATA, and another site here, which is where the friend leukemia integration site happens to reside. And sure enough, when we sequenced through these sites in a number of families, what we found were people who had precisely this deletion here in the enhancer and GATA box, and then multiple families that have single base mutations right next door to this in the FLY1 binding site. And these lead to monoallelic cis mutations that have contributed to disease that's identical to those other diseases. Now, we can take those mutations, we can put those into expression constructs, and I won't go into detail, but simply to show that when you put these constructs using the intronic element in front of a luciferase vector. The wild type gives you this normal level of expression, but when you take mutations that are either the 28 base deletion or that single base mutation, and you put those in the same construct, you can see that expression is diminished. But remember, when you're looking at a human, you're looking at somebody who has one perfectly normal allele and one mutant allele. So if you take those as percentages of normal, this is 50% of their function, and here they've got another 20 to 30% of function. But these people are diseased. So what that tells you is that you have to have something beyond 75% of normal function in order to be clinically normal. That is, once you get down to one normal allele and one mutant allele that is hypomorphic, the message being made off of this is completely normal. Those people still get disease. The implications of this are really quite profound because they say that the regulation of this gene is exquisitely sensitive and that it plays out over decades. So in these intronic mutations, the RNA is completely intact, but the levels off of one allele are low, and they still get sick. And so, as I mentioned, you need these features. Now, trying to understand these at a functional level, we've gone to what's called a nanostring approach. This is really just, you can think of it as a scaled-back microarray that is cheaper and easier to analyze, but if you choose your genes right, it's relatively well-focused. And when we look at this, we can begin to quantitate levels of GATA2 transcriptional activity. And here what you see is, regardless of whether your mutation is in the single base or in a missense allele, that the number of transcripts is significantly lower than here in the healthy control. So all of these people who have mutations of one sort or another have diminished GATA2 allelic expression, and we think this is what accounts for their disease. Now, let me just go one step further, because when you see someone who is phenotypically a disease, and that's of course what all of us as physicians do, when you see phenotypic disease, now we're in the mode where we've got to go and look for genotype. That is our mantra now, that we want to see phenotype, we want to connect it to genotype, we want to know what to do. Do we transplant? Do we treat with X or Y or Z? But what do we do in people that have unknown mutations? That is, that we could swear have our disease but don't have mutations. And for some of these now, I think we have 3 or 4, what we've been able to do is to go back and look at B-cell lines that are expressing GATA2, and we've been able to ask, do they express both alleles? And if they do they express them the same? So let me just walk you through this complex figure for one moment. So in this patient, who just came into the NIH 2 weeks ago, she had here in her genomic DNA, you can see that she's heterozygous for 2 different bases on either allele in the GATA2 locus. Okay? And she's heterozygous at 2 different spots, here at this base and here at this base. That is, she's got 2 peaks which are of roughly equal height. You can see this is what the normal peaks look like where you're homozygous. Here she's heterozygous, so the peaks are half size. Well, that's at the genomic level. But if we take her cDNA and we look at that, what you see is she's only expressing one allele. And the other one, which was easily apparent here in blue, is now gone. That is, she's got monoallelic expression of GATA2. Well, you know, we found this on a shipped sample, and her phenotype was a bit bizarre, and we were nervous that maybe we had made a mistake based on an artifact of shipping. So when we decided it was easier to ship the patient than to ship the blood, we checked all over again, and sure enough, we found exactly the same thing. And she's got other clinical features that are entirely consistent with GATA2. So you see here that we can find now people who have uniallelic expression of GATA2 who don't have identified mutations. Presumably, these will be mutations that are in cis with that allele that's not being expressed, and we have some very good reasons to believe that. And so we're now looking further upstream and downstream to see if we can find regulatory domains. Just to prove to you that we can distinguish this and that this is not intrinsic to GATA2, another patient with GATA2 deficiency, you see here, has perfectly normal heterozygous expression in cDNA, just like our patient has in genomic. And then just to prove that this is really the same disease as what these other people have. We've used this nanostring approach, again, using EBV B cells. Now, you might say, well, wait a minute, EBV B cells, what are you, crazy? You don't have to answer that question. But the importance there is that if we use peripheral blood, because these patients have variable cytopenias, monocytopenia, NK cellopenia, CD4 lymphopenia, if we use peripheral blood, we won't have any way to adjust between people that have normal counts or abnormal counts. So we have to find a way to get back to a target that can be used across platforms. And the EBV transformed B cell is as good as any, and it is renewable, so it makes it quite desirable. And when we look at our EBV transformed B cells and we do this nanostring array, here using 512 genes, you see that all of our patients, regardless of the type of mutation they have, all the patients cluster together and far away from normals or other patients who have similar infections but different etiologies. So GATA2 haploinsufficiency, I think haploinsufficiency of function and expression underlie all cases of GATA2 deficiency. And you can see you can get it either from exonic mutations, intronic mutations, or from people that are missing allelic expression that we're now calling uniallelic that underlie this disease. So in the last few minutes, I just want to try and remind you about some features of this disease. There are characteristic infections that Michael pointed out, HPV, herpes viruses, mycobacteria, and fungi, and it's an evolving syndrome, and that evolution is really what's hard to wrap our minds around. But as Michael pointed out, early on in life, they're pretty much normal. But remember, I'm an internist, so I know what you pediatricians think that means, but I'm an internist, so I only see patients later in life. And we haven't started to hear about all those children who show up with, leukemia, or show up with bizarre cytopenias, or strange B-cell disorders. And I would not exclude the possibility that there are some very early onset manifestations of this disease. You go back to the literature, there's a wonderful paper from St. Jude's of 3 children who showed up with B-cellopenia and developed myelodysplasia, showed up at age 2 and 3. And I would bet you dollars to donuts that those guys have this disease. And if anybody's here from St. Jude's who might have access to those samples, I am all ears. So early on, hematopoiesis is thought to be normal, and then they develop this progressive feature. The somatic features make this interesting. They get lymphedema, they get deep venous thromboses, and then the cancers, of course, are quite important. So this is what I sort of think of as the blind men with the elephant. We all see our own angle on this. Myelodysplasia, alveolar proteinosis, mycobacterial disease, wart disease, lymphedema, fungal, acute leukemia, all of these are manifestations of GATA2 deficiency. As Michael pointed out, the lymphedema, it's quite dramatic. And you might say, God, how does that fit in? Well, it fits in elegantly. In fact, when Stu Orkin first cloned this gene, he cloned it as an angiogenesis enhancer factor, and only later got interested in it being involved in erythropoiesis. And in fact, when you go back now and look, where is this gene distributed? One of the many places it's distributed is right here in lymphatic vessels. So here's GATA2 staining. This is a lymphatic valve, and GATA2 is critically involved in lymphatic poiesis, and vascular development, which I think is why they develop lymphedema and probably why they have deep venous thromboses. Lastly, I'll just touch on this because I think Jordan is going to go into in a little more detail, but this disease has really been around a long time, and it's only lately that we've begun to put it into its proper genetic heading. This paper from 1989 that really put NK cells on the map as critical factors in viral susceptibility, here published in the New England Journal from Christine Byron, Kevin Byron, and John Sullivan. And in this unfortunate child, she was 13 years old when she showed up with severe varicella virus infection, covered with chickenpox and varicella pneumonia. Remember that the patient that I presented to you at 12 had severe varicella virus infection leading to a 3-month absence from school. And after she recovered from her varicella virus infection, but not before that, she had problems with recurrent leukopenia and developed progressive myelodysplasia and ultimately died. Well, recognizing that this disease had a lot of problems that were similar in terms of the herpes virus susceptibility, I contacted John Sullivan. He was able to find blood from this girl, and we were able to verify that, in fact, she did have a mutation in GATA2 that was entirely consistent with this disease. And the being able then to go back and look at blood from other patients and say, well, really, is there an NK defect? And having outstanding collaborators like Jordan and Emily Mace, we were able to send them blood and find that patient blood, here in blue, has dramatically impaired NK function and NK killing. As you can see here, patients are really just unable to do what they're supposed to do, as opposed to normal killing, which is quite effective. And their NK development is impaired. And so NK cells are supposed to go through a process by which they go from being bright to dim, certainly what happens in aging all the time. And in fact, these patients' cells had problems in exactly this way. You can see that in control cells, there should be a significant population of NK cells that are bright, and then a larger population that's dim. Our patients had 0 CD56 bright cells at all. And in fact, if you took CD34s from these patients and differentiated them in vitro and asked, can we make them produce CD56 bright cells, the answer was no, not a bit. Here are the CD56 brights and controls, and here are CD56 cells from patients. So an intrinsic NK block that is associated with NK cell dysfunction and this problem in CD56 bright staining. Now, despite the fact that they don't differentiate normally and they don't function normally, if we treat some of these patients with severe refractory warts with interferon alpha, in some of these cases we've been able to actually restore some amount of activity. And so you see here one of our patients before treatment. Here's normal. Here's our patient, really impaired NK function. And here after treatment, you see function has improved, and both without and with IL-2. And I can tell you that clinically, their warts have really regressed quite dramatically with interferon alpha supplementation. So despite the fact that they got better, you can see that their CD56 brights did not come back. So these patient cells are still dim but working, things that have been said about me in the past as well. Now, I just want to come to the end here by reminding you that phenotype can really be confusing. And it's confusing because not everything is quite what it looks like. So keep in mind, as Michael pointed out, the phenotype can be quite variable. These 3 children at ages in their early to mid-teens all developed myelodysplasia, trisomy 8, severe viral infections. 2 of them had deep venous thromboses. All 3 of them had bone marrow transplants before they hit age 20. All successful, all doing very well. But their father, right, one of their parents has to be a carrier. There's no way that happens by accident. And their father is a 55-year-old hockey player who is doing otherwise pretty well, despite what you might think about hockey players. He's doing fine, despite the fact that his marrow is not normal. So at 55, he's asymptomatic. His marrow is not asymptomatic. We don't know what to do about that. Likewise, there are going to be many manifestations of this condition. And so this 12-year-old Afro-Caribbean boy came in with severe aplastic anemia, was seen on Neil Young's aplastic anemia service at the NIH. He got standard therapy for aplastic anemia with immune suppression and did very, very well, returned home. But 2 years later, came back with myelodysplasia and monosomy 7. You can see his bone marrow is now chock full of CD34 positive cells. He's got some of the characteristic bone marrow features of GATA2 deficiency with some abnormal peripheral blood neutrophils, and in fact developed acute myeloid leukemia and died. So he started out as typical aplastic anemia and ended up as GATA2 deficiency. Now, all these things make you wonder what, in fact, is coming first. What is the chicken here, and what is the egg? What's driving these problems? And to do this, you have to go back to experimental subjects that readily give informed consent. And so let me just show you what we did here. It was a very simple experiment. we took mice that either had mutations or did not, and we injected them with mycobacteria. Before they were injected, their bone marrows were completely normal and indistinguishable in every way. They were completely healthy before we got to them. But after 16 weeks of mycobacterial infection, and not before that, but after 16 weeks of mycobacterial infection, you see that the wild-type mice, who did certainly get sick, as wild-type mice do, but they had now got very lush and enthusiastic marrows, and you see they had no mycobacteria. In contrast, the GETA2 mice now have marrows that look just like what we see in our patients who show up sick with hypoplastic disorders. You see that their marrow is now partially depleted, and there are plenty of mycobacteria there. Driving home this point even further, if we take those mice and we dissect out their marrows and we say, do they have precursor cells in the marrow? Do they have stem cells in the bone marrow that we can find? Here are our wild-type mice, and at 16 weeks after infection, they've got easily detectable KIT-positive, SCA1-positive cells. And in all of these are heterozygous knockouts with one of these intronic mutations. In all of our heterozygous mice, you see they have virtually no stem cells left. That is, infection has driven their hypoplasia and their marrow exhaustion. So how does GETA2 do this? It's got regulatory effects in numerous tissues. It's got not only hematopoietic effects, but vascular and lymphatic effects. There are many overlapping targets of GETA factors, and I think this explains why the phenotype is so broad. In experiments I haven't shown you here that have been worked out in mice, they have diminished numbers of stem cells at birth, but they, I think, get consumed over the course of life with infections and perhaps other stresses as well. We think that at least in animals there's evidence for infection-driven bone marrow exhaustion, and we're now looking to see if we can identify neoplastic changes and cytogenetic abnormalities that accompany that. As Michael pointed out, the secondary mutations and cytogenetic effects are very important and are part of what leads to the development of the leukemias, and I think helps us know when it's time to go to transplant. So why should you worry about GETA2 deficiency? You know, like me, if somebody had talked to me about GETA2 defects several years ago, I would have just said, boy, that's a really great problem for a hematologist to worry about. I have more important things. But in fact, I would have been way wrong about that. Because these patients, you have to think about this disease proactively when people come in. Because these people go on to develop leukemia, just like the patient I presented you today. They get severe infections, not all of which are survivable. They get other cancers. The genital cancers in this disease, especially in young women, are absolutely heartbreaking. And I think preventable and treatable, but not if they're caught very late. Things like breast cancer and other solid organs, we don't understand how they're related here, but it may be partly linked through the NK cell function. And you have to be very careful. I saw a guy in clinic several weeks ago who at age 13, his brother had been diagnosed with AML and had received a bone marrow transplant. But they took the bone marrow from his identical twin, who was normal. But he died, the recipient died 6 months later. And the twin is now in my clinic 15 years later with severe GETA2 deficiency. So you've got to be thinking about this when you start talking about people who have bone marrow disorders who go to transplant that you don't have a good etiology for. And, as I've pointed out, the defect can present at different times. So a child who has this problem may have a parent who has the problem in whom it's inapparent. The argument of, they're not sick so I don't need to do it, is not a good argument. So when should you think about GETA2 defects? Well, I mean, there's breakfast, lunch, dinner. But in addition to that, severe viral infections, anybody who's got really bad warts that don't go away, bad HPV, and again, it's a recurring theme. When a 24-year-old woman comes in after her first vulvectomy and that, says, gee, but I've had these warts for 6 years, and nobody said anything other than, gee, that's just too bad, you got bad warts. Something's not right. And they come to immunologists eventually, and you have to think about what are the things that cause bad warts. This is right up at the top of the list. Bad primary VZV, I have yet to see somebody develop shingles. which tells you that their memory functions must be okay, but their primary functions must not be. And then bad HSV and really bad genital HSV is something we've seen in this disease. EBV, where it doesn't belong, the smooth muscle tumors now have been seen in several patients, and we have a patient right now with hydroa vacciniforme that I think is related to this as well. Of course, the disseminated non-tuberculous mycobacterial diseases, this is what got us interested in the first place. And then many of these patients start out with some pulmonary involvement, but that usually goes away, and then it comes back disseminated. The hypoplastic marrow disease, so myelodysplasia in general is not a hypoplastic disorder. And you have to make sure to go back and read that bone marrow report because those hematopathologists aren't interested in telling you everything that they found. It's their secret society. You need to break in. So having myelodysplasia is not enough. You need to know, was it hypoplastic or hyperplastic? Aplastic anemia, we've now seen several cases of people who were called aplastic anemia who turned out to be this. Neutropenia, 10% in a French registry of kids that were called neutropenia turned out to have GATA2 deficiency. And then monocytopenia and B or NK cell cytopenias. Finally, severe disseminated infections with fungi, histoplasma, cryptococcus, soft tissue infections of aspergillus that I've never seen even in chronic granuloma disease. These people get big chunks of flesh in, say, leg or arm with aspergillus. And then we've seen a few cases of severe dermatophytoses, not the sort that have been reported in CARD9 or STAT1, but mostly on skin, not mucocutaneous, but really only cutaneous. So GATA2 deficiency, I think it's much more common, certainly more common than I ever thought, more common than we have identified, has multiple manifestations. It's got a variety of infectious manifestations, so it's not going to be easy to corral as one simple defect. It's got variable onset, progressive cytopenias, haploinsufficiency. So this is a fundamental regulatory gene problem. And then I think, as Jordan will point out, it's really got an interesting angle on how NK cells do what they do and how important they turn out to be. And I am here to tell you that it took me a long time to figure out just how important those NK cells were, and kudos to Jordan for persisting in the face of skeptics. So this GATA2 is really one critical gene that unites infection, hematopoiesis, immunodeficiency, lymphedema, and cancer. So just remember, things change over time. You want to really be careful about the sun down here in Miami. You can see what it does to the unwary. So GATA2, think about it almost all the time. And I'll just acknowledge people that have done all the work I've shown you here today. You've heard Michael Spinner's outstanding performance over the last year, putting all this together, and our genetic and cellular group, as well as our collaborators around the country and throughout the NIH. Thank you very much.